Hi everyone, it's Dr. Renee again, and I'm bringing you important news from Sigurd Hospital concerning a new experiment that was done. The experiment aims to change the way that Adenosine Deaminase 2 deficiency, a disorder, can be treated in the future. I'll explain what the disorder is, how the experiment was done, the results, and why it matters to you. DADA2 is a rare autosomal recessive disorder, meaning that both parents might have a mutated gene. In this case, the gene is ADA2 is believed to make the enzyme Adenosine Deaminase 2, which is thought to aid in the development and growth of macrophages, where cells to destroy foreign molecules and induce inflammation. When the gene is mutated, it will cause the macrophages to release cytokines, which are proteins, and some of the cytokines are interleukin-1, 6, and TNF-alpha, which are associated with inflammation. Also, there'll be excess inflammation, high blood pressure, liver, spleen, kidneys, and gastrointestinal problems. These disorder can be identified through genetic testing, and there's currently no cure. Some treatments given to individuals are anti-inflammatory medication and phototherapy. Some are given human stem cells to make these cells in the body reintroduce the enzyme activity that reduced by this disorder. To begin the experiment, researchers spotted human bone marrow, human macrophages, and mice, and they had to get approval from the government bodies to test on these tissues. The human bone marrow cells were then isolated, purified, and washed using a CD34 microbead kit. Then they were placed in a medium that contained many different compounds to allow these cells to grow and develop for 22 hours. Once the 22 hours were done, they were injected with lentiviral vector LVADA2, which was the type of vector that infected cells which contained the functional ADA2 genes. Transfer process was done until the multiplicity of infection tests indicated that the virus to host ratio was sufficient enough. After 16 hours, the cells were gathered, washed, and placed on nutrient supplement plates to grow for 14 days. Then these cells were placed on such plates to evaluate the number of colonies that formed and how they looked. The process was used to implant the LVADA2 dog mice. Dog mice were made by taking out the ADA2 gene. Using the CD14 microbead kit, select macrophages from patients were purified and isolated according to the manufacturer's instructions. These cells were then placed on nutrient plates alongside colony-stimulating factor to promote their growth. These cells were then infected with viral protein VPL, VPX to increase the expression of the LVADA2 introduced by the Lyme vector, and they were allowed to grow for overnight. We performed the macrophage polarization process in which macrophages change their structure function in response to microenvironment stimuli. These were treated with Escherichia coli liposaccharide to increase the enzymatic ability of the macrophages. They are also treated with human recombinant interferon gamma to stimulate the antimicrobial and anti-tumor activity of the macrophages for 48 hours. When analyzing the data for the knockout mice when they were reintroduced with the ADA2 gene, it was seen that they had increased levels of immune cells in plasma and maintained a healthy weight, and they maintained also a healthy inflammation response, and that they had no transplant rejection of these cells. Additionally, when they examined the effect that DADA2 had on bone marrows using flow cytometry, a technique that examines the physical and chemical characteristics of cells, they found that there was a cascade of immunocellular problems within the bone marrow cells. Additionally, overall, they found that the ADA2 gene reintroduction inside the bone marrow and the patient macrophages was successful, since it reestablished the enzymatic activity within them, allowing for a healthy immune response comparable to a non-deficient healthy cell. Although the results from this experiment seem to be very promising, more research and experiments need to be done to confirm their validity. Nevertheless, this doesn't take away from the notion that these results can lead to a more improved clinical treatment for individuals that face adenosine deaminase 2 deficiency, since the treatment seems to have almost no side effects and more benefits compared to other treatments on the market. As a result, this can lead to individuals having a better quality of life. Lastly, the results and message from this week can hopefully help other researchers evaluate how they conduct their own research concerning disorders that affect the body in a similar manner. Thank you for listening to my presentation, and I hope that you find it to be very informative and you want to, you can reach out to me to find out more.